technologies. Now, our lab at IIT Bombay is about 15, 17 years old this year, but all this translational work started happening somewhere around, I'll say 2010, 11. First 5 years is most professors would spend their time, making their paperwork strong to get promoted, and that is what I was also trying to do. So 2005 to 2010 is when most of my students were there in the lab. They were working on their PhD thesis, they were working on MTech thesis. We had many, I'll say, funded projects. But somewhere around 2010, I realized that this is not the trajectory I would like to go. I would like to make it different. And when I thought about different, I thought I'll try to do everything on a large scale. I'll train as many students as possible. So I had about 24 to 25 PhD students at a time in the lab. I will have, maximum number of masters in the lab, MTech students, about 7, 8 MTech students at any time. I will have a lot of funded projects, lot of staff, lot of postdocs, and most importantly, I'll give training to several interns. These interns are the best because you get a lot of students who would like to come use uh, the IIT facilities, sharpen their research skills, and then move out into the real world sort of research world with our recommendation. So we get a lot of these interns, and I had many of these come to our lab. At this moment, I have about 35 interns in my lab, and about, I'll say, 20 PhD students, maybe 7 or 8 masters, and several postdocs. About 60-member to 70-member lab at any time at IIT Bombay. Now, why I'm saying this to all of you is I just want you to realize that with the facilities that a government organization like an IIT or maybe any other institute offers to a faculty, we have the flexibility to do more or less. I chose the more path, and I'll show you how that has been helpful to not just me, but to many of my students, to many of the interns we trained in the lab. So once again, warm welcome to all of you. My name is Rohit, and I'll talk about what we have done at the Nanobios lab at IIT Bombay. If you look at the ecosystem at IIT Bombay, it is typically the standard ecosystem that exists at most of the institutes in this country. You have several departments, engineering departments, the ones in dark blue, chemical, electrical, mechanical, industrial design. You have several of these science departments, biosciences, chemistry, computer science, engineering, physics, or, and material science. So many of these science, engineering combined, you have skills across these two departments. You have many labs in these two departments, in many of these departments, which have a focus towards translational research. And then you have many of these things coming together, like you can apply for private funding from several donors, several of these large, I'll say, funds, trust funds, or you can get public funding like DBT, DST, ICMR, and whatnot. Today there are many of these funding agencies, and they're very, very, I'll say, efficient in giving money. Then, most importantly, if you're trying to do healthcare research, you should have a tie-up with a medical institute. And that is something that I consider crucial, because none of us have a medical college on campus, except IIT Kharagpur, and now there's one coming at IIT Kanpur. There's one coming at IISc Bangalore, but none of the other IITs have this liberty, which means that you have to have a tie-up there. And of course, you can tie up with any incubator, you can tie up with R&D laboratories, but if you use the combined skills of what you have on campus, use the current ecosystem that you have on campus, existing ecosystem, which can take an idea all the way into the market through these incubators, you now have a system in which you can set up your startup company through your own students. We've done that. Or you can transfer the technology to a medtech industry. We've also done that. But most importantly, what you end up doing is making these indigenous products, contributing to affordable healthcare in India, and doing capacity building in this country. So our department at IIT Bombay has about 29 faculty currently, several postdocs in different labs, many master students, but most importantly, a large number of PhD students who are the workhorses of the department. Still don't have a UG program, but we are working towards getting a UG program approved by the Senate very soon. There are many challenges of the medical device industry. I don't need to tell you all this, but most importantly, please focus on the top right hand corner. what you will see is these different countries, Brazil, Russia, South Africa, compared to India. And what am I comparing? I'm comparing health expenditure per person. So India spends a very low amount, $75 per person. But how much of that is private and how much of that is public spending? And look at the numbers. 70% is private spending, which means even with the small numbers that we are spending, if I cannot afford that small value, it is of no use to a normal, or the population of India. So with that one thing in mind, we started focusing on probable solutions, and we thought we'll work on affordable healthcare technologies. And I did not want to just work on devices. I also wanted to work on, therapeutics, on nano, on on technologies, across the spectrum, so that I could actually contribute to what this country needed. And we did many things over the years. So some of our major research areas are photothermal therapy for cancer, magnetic chemohypothermia, and these were the ones that I originally started or initially started when I joined IIT Bombay, because I did not want these research areas to be stopped. I actually wanted them to go forward, and these take time, as you will see. But then there were many other things we started, like affordable paper-based diagnostics or microfluidic-based diagnostics, or biosensor work or point-of-care diagnostic work. This work actually moved faster. So there are two of these streams. One stream which moves slower because they have many hurdles to cross before they reach the market. One which can reach the market fast. We have many new areas which got added to our lab, consisting of protective care equipment during COVID times, disinfectant sterilization products, drug-eluting devices, IoT-based health solutions, and many such new areas which actually augmented what we were already doing, plus added new dimensions to our lab. We have commercialized many technologies. As you can see from this circle here, many of these awards that I got are because my students set up these startup companies, actually took an idea all the way from an ideation stage to the market. And Biosense Technologies was one such company, which took ideas like the U-Check, which was his master's project, the Sync, which was a glucometer, the TouchB, the Smart Sense Analyzer, and many such ideas, all the way from a very small, ideation phase all the way to market. And there are many other companies which joined hands, with us over the years, and many of these are student companies like Dynasense, like Mozen Technologies, Kavach Bio, CareNX, Patanjali Pharma, Neodox, Norditech. Many of these are student companies, or even interns who worked in my lab then set up companies, and they actually put me as advisors in these companies. So that's a lot of benefit the lab got from having been associated with these students, with these staff, these interns. And many ways IIT Bombay also got to use its powers of the ecosystem to actually move things forward. So we've commercialized, like I said, we've commercialized the U-Check right there in the center there, which was a master's project for a student who set up a company and took it all the way to the market. This was the first example of a mobile phone-based diagnostic device. The one on the right-hand side is our newest model of the glucometer, which went through several iterations before being commercially successful in the market, and it's called Sync. And it has strips which were available in the market at 5 rupees, compared to 18 to 30 rupees per strip, which was available even during that time from large multinationals. The TouchB, which the students company commercialized through their efforts, is more of a screening tool for anemia detection. And this actually made a big difference to the country because it doesn't use blood, it doesn't use any invasive techniques, it actually uses non-invasively or just the conjunctiva of the eye to tell you the hemoglobin levels. There are many other technologies. We have worked on technologies. Like I said, we've got a platform technology. Now, how do we add assays to the platform technology to create more and more point of care diagnostic devices? We've got Smart Sense, which is point of care device for doing, sodium, potassium, chloride, bicarbonate, pH, urea, glucose, and creatinine. So all of this at point of care. A point of care device for doing just sodium and potassium in a different way, so that this can be used in uh, uh, you know in the internal medicine purposes. We also worked on a URITSA device, which is albumin and creatinine ratio, and a device around that so that it can be used for kidney disease screening. There are many technologies we've worked on. We've got call check, which is our affordable cholesterol screening device. We've got a screening device for measuring glycated hemoglobin, HbA1c. We've got a screening device for measurement of vitamin B12 and D3. So all of this are many of these are under development. Many of these have already gone beyond development, have been transferred to a company. I'll show you some of these as I go along. But most importantly, what I want you to focus on is our lab does across the breadth of biomedical engineering work, which doesn't involve one vertical, but all verticals of biomedical engineering, which is crucial. When you are in a department like biosciences and bioengineering, you can actually use the expertise of each and every faculty in the department to work collaboratively, to actually be able to take an idea all the way from your head, which is ideation phase, to the market using expertise that exists within your own department. One example is our affordable novel biodegradable bone screw or tissue fixation devices or materials, which can actually be used in an in vivo condition, rather than all of these devices, many of these devices which work with blood or urine or sweat, in that matter, which is a non-invasive sample. In this case, this will actually go inside the body, so there's a longer route to reach the market. But we've set up a company. I'll talk about that. So we've done many. Most importantly, please remember, if you're working on a healthcare product, do not make the product and then go and find out where it will fit. First, find out where it fits, who will need it, what price point they are agreeing to pay for it, and whether you can actually penetrate the market with that price point. If you can do all that through a case, through a questionnaire or whatever you float to these people, you'll be able to make products that will be taken up in the market. If you make products without looking at what the market needs, you will find that you have devices, you have assays made, but nobody is wanting to take them forward. So this is something that we never do. We actually figure out what the market needs. And in this case, we actually independently surveyed these 50 path labs. We found that 30 of these did not have a semi-automated urine analyzer. And the ones that did were very, I'll say, careful in using strips, because the strips were expensive. So one, we had to remove their dependence on Siemens and other strips, which were expensive. And the other, we had to introduce a reader to them that could actually be used in multiple purposes. So we thought of working around that problem, and we got a funded project from DBT, the IYBA grant. And actually, we started working on a device that can reach the market using a mobile phone diagnostic platform. It's a very simple platform. This was the first example in 2011 when it was made, about 11, 12 years back, when mobile phones were just becoming really famous and in everybody's pocket. But they were still expensive at that time. Nobody was thinking of using mobile phones for diagnostics, at least. Today, there are many such examples. But most importantly, you can use the camera of the mobile phone. You can adjust the lighting if you put it in a right box. And this is what the box is shown as. And you can actually take an image of the strip as it changes color, or keep taking multiple images at different time points. When you do that, you can compare these images, or the RGB values of these images that you take, to inbuilt calibration curve, and then give values to these pads. What are the pads? Leukocytes, bilirubin, urobilinogen, protein, pH, specific gravity, and so on and so forth. This is the 10 parameter urine dipstick. Then there's a 10 plus 2, which is albumin and creatinine. We worked on both of these and compared our device to what existed in the market. Our device was costing about 15,000 rupees compared to a Siemens reader, Clinitech, which is costing about 75,000 rupees. And our strips were costing 30 rupees compared to 100 rupees Siemens strip for the 10 parameter, or 80 rupees for the 10 plus 2 parameter, which is Siemens strip is costing about 250 rupees. So if you can look at the market dynamics, if you can look at where it fits, you can actually create a big market. So the company made crores of sales every year. Finally, with all the sales that they were making, they were taken over by PerkinElmer. So I think 4 years back, they were taken over by PerkinElmer in a very large deal. and today they are part of PerkinElmer, which is a very, very big success story for the students company. They also worked on a self-monitoring blood glucose system. This is part of the panel that I was in New Delhi, and ICMR came out with uh, its own indigenous glucometer plan and said that whoever makes this will actually be able to commercialize this in India. And there was this big plan. We looked at the market, we figured out what is needed, and most importantly, you figure out where you will get hit by a patent infringement. If you think that there is a patent infringement on the technology, don't work on it. But if you think that you can work around the patent, or you can wait for a couple of years for it to become open access, then just go ahead and work on it. By the time you're ready to launch it in the market, you will actually have it as patent free. So the blood glucose monitoring is a simple technology. It's a glucose oxidase peroxidase method. It is something that you read in the books. It is not something that I have made or somebody has made. So nobody has a right on it, which means that you can use it or anybody else can use it. Nobody requires a patent for it. And you can make a strip for it. We made a strip, and these were color monitoring strips. And with this strip and a reader that you make around it, we actually could be able to read glucose readings, in blood. It had to be approved, which means that it had to go through a strong validation plan. And we went through a strong validation across hospitals in India, supported by ICMR. And we got 95%, more than 95% of our readings within zone A of the Clark's error grid. This is the most standard way of testing a glucometer. Once you can do that, then you'll get approval to sell it in market. We got our manufacturing license, started selling in market, made a big impact, and slowly started working on different versions of it. The most recent version does not use calorimetry. It uses electrochemical strips, which are much easier to manufacture for that matter. But most importantly, it was developed and marketed as part of a plan funded by ICMR through IIT Bombay and Biosense Technologies, and released in a very large function in New Delhi, where the minister released this, and it made a big splash. And most importantly, it created a big name for the country to have come out with its own indigenous glucometer with strips which were costing 5 rupees. We worked on many technologies. Smart Sense is one, and I say this to all the students. Start looking up awards. Start looking up these these, I'll say, competitions which are being held everywhere in the world for healthcare. And I'll give you one example. This is the Healthcare Innovation Technology World Cup, HIT Labs World Cup. In this World Cup, we had applied, we got selected, we went through several rounds of screening. Finally, IIT Bombay, IIT Kharagpur, and there was another team. So we were the top 3 teams in the, and there was a team from, I think, from US, I think MIT. MIT team was first, Kharagpur was second. We were third in that final competition of 160 countries. And finally, 3 of these, the top 2 were from India. So you can imagine, you can make a difference. The only thing is you have to have a strong idea, and you have to have the will to push it through. If you do that, any idea, however small it is, can become a big idea. So we were able to push this. We got it funded by Wellcome Trust, went to London, presented this to Wellcome Trust in London, got it funded by their scheme, which is Affordable Healthcare Technologies in India, and started working on this. The idea was to make a critical care diagnostic device which sits in the ICU and is able to do everything at the bedside, which is why you see their centrifuge on the side, because most of these cases you'll never be using whole blood, right? You will draw whole blood, but you will always be using spun down, which is either plasma or serum. Now, it depends on what is being done in the hospital path lab. You are trying to do exactly the same thing at bedside. So you're not going to change the way. And the way we chose this was, we had pre-filled cuvettes with our reagent in it. All we had to do was add the serum to it. In some cases the serum, in some cases plasma, depending on what you think is going to be used in the hospital path lab. And we were able to then compare it to the hospital data and show that we had very little variations, and most importantly, we had an assay which was really cheap compared to what existed in the market. So we then transferred the technology. Then we also worked on a kidney-related disorder, early detection of kidney disorders, which is doing albumin creatinine, not using the the strips that I showed you and the mobile phone, but using a separate reader. And this we got funded by DST. Uh, Completely different technology using colorimetry, where we are going to measure the absorbance, of these two different dyes, when they come in contact with albumin and creatinine. And as you can see, the albumin, you are going to see a change in color. With creatinine, you're also going to see a change in color, and there's a stark difference in color which is able to be picked up by your reader. And once you can do that, you'll have to work on different grades paper, on which paper you have to put it, how much dye you have to put in, how much of the buffer you have to have, how much dry you have to keep it, and what conditions you have to store it before you can use it. So once we did that, we made a reader around it, and this everything was done in-house in our lab, where we have the capability of making our own PCBs. Of course, you can design the PCBs, you get it printed elsewhere, but you can actually work on the electronics for the device. You make a device, and this is actually a video of the device which shows how the device works. So you can see that actually it can measure all uh, the albumin creatinine ratios. I'm just running through the slide so that you can actually see values which are coming up as the device can measure albumin and creatinine. Sorry. So let me go to the next slide. So we've got many such technologies. This is just the top view I'm showing you. We also work on microneedle patches for transdermal drug delivery. Now, this is going to be a huge technology in the current times because of vaccine delivery, but we are hoping that we can work on many such technologies together. And you can actually use expertise of mechanical engineering, of material science, to get materials, to look at fluid flow, fluid dynamics, and look at materials that can degrade slow or fast. And then you use the expertise of biosciences, bioengineering, to look at biodegradable materials, and then drug delivery. So we worked on such technologies. We've got technologies that are available for licensing from our lab now. Many of these you see at the top there, you've already transferred them to the company, or we are working in close collaboration with a company to actually get it to market. We've also got a cholesterol, total cholesterol, HDL, LDL cholesterol, and triglyceride assay made along with a reader. So once you make the assay, and these are the slides which show you the assay, and these are all colorimetric strips. If you can make strips, you can make a small liquid reader for that as well, because everything works in liquid phase as well. So you can work on two different readers. You can work on a solid strip-based reader. You can work on a liquid assay-based reader. Once we design these, we prototype them, we make the readers, and then we start testing. So we look at stability of the assays, we look at stability of the readers across different conditions, and then we actually do clinical trial testing, which is samples from hospitals after approvals, ethics approval. So once you do that, you compare your data to what the hospital's data is and see how much deviation you get between the data. If you see less than 10% error between the data, then it's good to go. Then you can go to the next step and start working on a test license, so that you can then do 3 batches of 100 samples each. And then once that data is submitted to CDSCO, you can actually come back and get a manufacturing license. So that's where we are at. We went ahead and got a BIRAC CRS grant for doing a scale up, for doing a validation study at 3 different hospitals or 4 different hospitals, and finally making manufacturable strips. So we've got a reader now, we've got strips made, and we can do all of it in the same reader. Plus we are making an industrial design for the reader for the liquid assay. So once that is made, then that will be good to go. And we've also got our manufacturing of batches done. We've got our test license approved by CDSCO, and we will start testing at hospitals. So that's where this grant is at. But I'm hopeful that by the end of this year, maybe beginning of next year, we'll get this product in the market. We're also working on a detection of glycated albumin. Now see, you can work on a spot glucose, which is for diabetic patients, but there are 2 other tests that are good for diabetic patient. One is a 3-month marker, which is glycosylated hemoglobin, which is HbA1c, and then there is glycated albumin, which is another marker which can give you a 3-week glucose reading in the blood. So if you can actually make these assays, you can make a full glycemic panel and then put it in the market. So people who are diabetic know that they have to test themselves daily. People who are prone to diabetes or have familial diabetes, family diabetes, they can actually test themselves every 3 weeks. people who are on the borderline can actually test them every 3 months, and you can actually create a big market for these technologies. So we started working on this. Of course, you have to think about where and which market this should fit, and we've got our own assay. Again, like I said, you have to work on an assay for it. Make sure you have to have a reader for it, and then make sure that your assay and reader work together to actually give you the readings that you want. Then test it in a hospital setting so that you can test it on real samples, not spiked samples or buffers. You have to actually test it on real samples, in this case blood or urine or whatever you are trying to test it, so that you can compare your data. Then go to CDSCO and get a test license. So uh, again, all these cases have, we've made our own centrifuge. You can actually spin down the blood at the site, and once you can do that, you can actually take the plasma out and start working with the plasma. So sorry. So there are many things, we're working on getting that technology in the market. Again, there's a funded project for that, but we've already transferred the technology out. We are working on several affordable healthcare products using nanotechnology also. So not just simple, diagnostic assay work, but also complicated work which involves drug delivery, which involves photothermal therapy, which involves actually using other pillars of bioengineering in order to get what we would like to get. So we got our gold-coated nanoparticles for photothermal therapy in cancer. We got our nano-engineered biodegradable bone and tissue fixation devices. We've set up a startup around it, already got funded for it, and we are now moving full on to start manufacturing and doing clinical trials. We've got our own drug-loaded nano-engineered sponges to fight periprostatic infection. Again, we've got a startup around it. And we've also got a nano-engineered sponges for on-demand drug delivery, for which we've transferred the technology. So the many places where I say, we've transferred the technology, there are companies that are actually interested in taking your technology. You should have a technology either at TRL3 to TRL5. When I say TRL5, which means it has already completed clinical validation, lab scale clinical validation. But when I say TRL3, it is, sort of in the early stages of validation. Okay. So we got many of these. Like I said, I'm not going to go through them. Photothermal therapy is going to be a big technology in the future, and I'm sure we'll be part of that. We are setting up a startup around it, and we are hopeful that this will soon reach a phase 1 trials, with the GMP manufacturer that's being set up at IIT Bombay. This is a liposome technology, which means there is a liposome core which is biodegradable, because it's a thermoresponsive lipid material, and then there is a gold coating on top, which is a shell-like coating, which is created out of small nanoparticles, sub 5 nanometer particles, which creates a shell. Now, with the shell and the lipid material, you are creating a dielectric medium through which you can actually send light in and convert that light into heat using surface plasmon resonance. Once you can do that, you can use the heat to kill the cancer cells on site, and the material itself degrades and is excreted from the human body. So there's a lot of work that has gone into this work. We've published a nano letters paper also, and we've made sure that we've got all our preclinical data ready in order to float the company and take it to clinical trials. A lot of good work being done with IIT Hyderabad, one of my students, Arvind, and at Actrek with Abhijit Dey, and our own lab at IIT Bombay. So several of these people have come together to make sure that there's a technology that moves forward. We've got many other technologies. Rajinder has worked on many technologies in my lab, and he's now applying for faculty positions across IIT. I'm pretty sure, with the work he does, he will be successful. We got our own bone screw. We set up a startup with Professor Bilare, his student Kunal, and one of my students. And we've actually got funded for $1 million already. And with that $1 million, we will be able to start manufacturing and taking it to clinical trials. Our screw is much better than what exists in the market from Smith and Nephew, which is a bio-RCI hyaluronic acid screw. And this one is PLLA HA screw that is much more expensive, 15,000 rupees per piece, compared to our screw, which you see on the top left corner, which is made out of these 3 materials at the bottom of your slide: silk, magnesium oxide, PCl. PCl is slow degrading. Magnesium oxide is going to give it some more properties like biodegradability, biocompatibility, antibacterial, and most important, silk, which gives it a little strength. So if you can make such screws, it can degrade and it can be used in vivo applications. It has a huge market in this country. We've got novelty, we've got feasibility, and we've actually several patents filed around this area with ICMR, not just Indian, but US patent, Japanese patent, European patent, and many others being filed because this technology has the potential to move further. The USPs are there, and we are pretty sure with the funding that we've received in the company, we'll be able to make a big impact. We've also worked on Navrakshak, uh, you know, mask in our lab, and this Navrakshak is a trade name brought in by one of my student who's a Indian naval, surgeon. He's doing a PhD in my lab. Imagine that, a Navy doctor who's doing a PhD at IIT Bombay, but just because he's passionate about affordable healthcare, he's already filed several patents in our lab. In the last 1 year, the two of us have filed 30 patents from our lab, and all these technologies now getting licensed to companies, so that there is a big inflow of into our lab of funds coming in from companies, but most importantly, there's a big inflow of royalty coming back to IIT Bombay with sale of these products over the next 15 years. And that's the goal with this. Many of these technologies already got this license. He's currently in New Delhi today in order to discuss how the the entire system sets in. We got smart wound dressings. This has been transferred to a company in Navi Mumbai. They're very interested in using micro needles and the patches we've made for faster healing of wounds, and in fact being able to deliver other materials through the needles, so that the wounds can be closed faster, and this can be used for diabetic foot ulcers, for many such wounds. We've got a lot of data around this, but right now we're working with the company to be able to do preclinical studies, so that we can actually take it further into trials. We've got on-demand smart wearable devices for pain management. Again, these are available for licensing, but one company has shown interest very recently. We've just signed the NDA with them, and hopefully the company will take this technology forward, where we're using sound wave or sonophoresis, ultrasound, to actually push the drug into the skin. This is not a novel technology from our lab. It is known technology, but then how can you use that for pushing a certain drug into the skin for a certain application? In this case, we are going to target osteoarthritis. We've got several of these materials which have been transferred to YBL as well. Many technologies we are working with. We are working on a micronutrient malnutrition screening device, which will have multiple micronutrients being made, assays being made for screening. And of course, we've also working on a urinary iodine screening device, as you can see in this slide. So many technologies. As you can see, if you work on one, there are 5 more that open up in front of you. You can have one assay made, maybe using the same platform technology, you can make multiple assays. So keep that in mind. There are many of these areas that one can work on. But most importantly, if you have one such product in the market, you have the confidence of bringing many more to the market. We've got our own T3 T4 TSH screening device, and we've made many technologies around it. We also work on biodegradable packaging material, because that's going to be a big area in the future, and we've made some of our own packaging material, as you can see in this slide. Now we are making spoons and and straws of biodegradable packaging, so that can become a big market. We've already got funding from IRCTC, and we are hopeful that we can take that technology forward. We do a lot of social initiatives from our lab, and this is including malnutrition screening. We've started doing this at several places in this country and the Northeast. And most importantly, we use what the students have made, the devices that the students have made, in order to set up these screening camps, or actually do it at Anganwadis, or actually do it at primary healthcare centers, using smart scale for monitoring, smart height measurement devices, anemia screening tools, or even pregnant women screening using the Care Mother platform that, again, a PhD student has set up a company. So we've got all these technologies in our lab. How do you use it for doing screening? And the Care Mother is an excellent example. We've saved many lives in the last 5 years of the company's existence. And today we are proud to say that many pregnant women we have saved. Most importantly, these mothers we have saved, and most importantly, their child has been saved, because we were able to flag these high-risk pregnancies when these Anganwadi workers went home to home using these kits, as you can see on the right-hand corner, and were able to screen these high risk pregnancies, sent the data to a primary healthcare center, and these were then referred so that appropriate interventions could be done. We've also done screening in camps in Rajasthan, in Churu district, and in several slums in Mumbai. We got data from districts in Maharashtra, districts in Rajasthan through the student companies, of course, and Care Mother. You can see how many of these mothers we have served and how many we have actually been able to save. We've got several grants awarded to Nanobios Lab, not just the BIRAC grants or the DBT, but we've got international grants, Bill and Melinda grants. We've got Grand Challenges Canada 2 times. We've got the Welcome Trust Award. We've got GYTI awards, which are a big award. I encourage the students to apply for this award because it's actually a very prestigious grant given by the President of India in March of every year, and this makes you work on a different scale because now the country is looking up to you for your ideas. We've got several of these impacts created by Nanobios Lab, not just the PhDs that we've been able to graduate. In 17 years I've graduated 40 PhD students, not just registered, graduated, and 20 more are registered with me. We've graduated 90 master's students, but many more are registered, so by maybe next year we'll have completed about 100 master's students. But most importantly, we train a lot of interns and we publish papers as well. So we've got more than 220, 30 papers now, international journal articles now. Good H index, not very high, I would say, but then we have created impact. In terms of training people, in terms of training interns, and also giving the right training to the innovators. Winning many awards, but then filing many patents. Our lab has filed more than 150 patents now. And last I counted, a few days back, it had already reached about 165 or so. But many of these have been granted. We got more than 30 now granted. Every week now we are getting one patent granted. That's the speed at which we are now being able to file patents in 4 years, 4.5 years, 3 years, 3.5 years. Our patents are getting granted, and we have a big chain of all these people who are wanting to now take the technology to us to a company. And every month we are trying to do a tech transfer. We commercialize ideas, we commercialize assays that we have developed, we work with companies, we actually create impact, and we have been able to create high value jobs and benefit patients. We get recognized, of course. Whenever you do good work, your students do good work, they are recognized at every platform. The Honorable Prime Minister was there, the HRD Minister, the Science and Technology Minister, you name them. Everybody has seen our lab, has actually seen the kind of work that the lab at IIT Bombay does. So I'm hopeful that one day all the students will be recognized for everything that they do. So I encourage each one of you to do what I have done or my students have done, so that we can create multiple such ecosystems in the country. Of course, we do a lot of outreach. We go to several places, talk about what we do, like I'm doing today. We encourage youngsters to do what we do. All the young faculty I encourage to do what I do. Don't take 4, 5, 6 PhD students. Double the number, triple the number, and see the impact that you're going to create. Train more people in your lab, not just PhDs and masters and BTechs, but also outsiders, because these are students who work very hard in order to give back a lot to your lab and to the institute. So do that, and I'm sure you'll all be successful one day. We've got a huge partner network, not just institutes, but hospitals, student companies, large companies, Indian Navy, name an MOU. Every week, our lab submits at least 3 to 4 NDAs or MOUs for approval by the Dean's office. And we are, at that some point, that was what our lab looked like, 50, even today it is about 55, 60 member lab. Most importantly, all this is possible because of all the mentors I've had. The most important, Dr. Manju Sharma, who is a big mentor to all youngsters in this country. Professor Padmanabhan, who chairs most of the meetings I have been part of, and he has always guided us in what we do. And Dr. Surup, who as Secretary DBT was instrumental in doing many things that this country needed. So, she has been a pillar of support to us, to many of the youngsters in this country. Finally, let me thank all my faculty colleagues, my mentors, my friends, collaborators, my students, and all the startups that I have worked with, all the companies that I have worked with. Thank you very much, and I hope that one day we will see many such nanobios lab in this country. Thank you.
Thank you, Dr. Rohit. It was very nice presentation. I am sure the students are quite motivated. I request students to ask questions.